All right. Well, it's great to see you guys. Thanks for joining us today. We're really excited because we have decided, you might have noticed that we've kind of mixed things up a little bit with our meeting schedule. And one thing, really that was based on feedback from the community about what they want more of. And we're trying to meet a lot of needs and trying to ensure that we're giving more families the opportunity to connect. We recognize that when we had meetings that were for specific variants, it meant that some families just weren't having access to meetings that really suited them. And so today what we're doing is we're having a research update, and we're going to do this 3 times a year at least. We're kind of feeling out this new schedule as it goes and seeing what you guys think, and we love feedback, so feel free to let us know. But the idea is that 3 times a year we will give these research updates, and Michael will talk about what we're learning from the data you all are providing in the registry. We'll talk about kind of some of the things that are emerging that we're seeing in the field. And just an opportunity for you to also ask questions generally about SCN8A and what we're learning about it. So before we get started, I just wanted to, I don't know if people regularly visit our website, but if you do, you'll see that we have right here, it says upcoming events, and you can always go there to find whatever is coming next. So here's today. We're really excited that we're also 3 times a year going to be hosting open themed meetings or open topic meetings. And the one coming up on April 1st is on inheritance and mosaicism in SCN8A. We're going to have our first meeting with Dutch families, building in some more newly diagnosed programming, meetings for families who just want to talk. So there's a lot of opportunities to connect here. So check it out if you haven't yet. You can click on any of these and then register, and it'll be on your calendar. A new feature we also have is that our website is now can be translated into about 100 different languages. So please, if you know families that need maybe information in a different language, they can now come here and see it in one of these 100 different languages. So I'll leave it there for now. And I want to really let Michael have the floor. I think towards the end, I can give a few updates on clinical trials and other things if people are interested. But for now, Michael, if you're there, I think let's jump in, because I know you've got a lot to cover and people are excited.
Well, good morning, good afternoon, good evening, wherever you are. It's great to see you all. So I think I'll just start sharing my screen right away. So the way I conceive this, and I'm open to suggestions, but as I was speaking with Gabi and Jada, it seemed like a good way to go would be to have a series where we cover different topics. Each time I'll try and say something about how the registry data and all the information we are learning from you is contributing to these different topics. And I just put these topics together as possible different ways of going forward. So I'm very much interested in hearing what you guys want to hear about. But here today, we're going to start with the DNA mutation inheritance. Then next time we'll talk more about, the order of these can be anything you like, but neurophysiology and epilepsy, like what is the neurophysiology, what's going on in the brain, the neurons, the circuitry, and what are different seizure types and where do they affect the brain and things like that. And neurocognitive development, which is probably one of the most important things that we're all concerned about, is how our children and loved ones develop and progress in life and what their capabilities are, and what we know from SCN8A and how neurocognitive development is affected by SCN8A. And then I know there's a lot of also interest in drugs, new drugs, old drugs, future therapies, And for this, we might ask for other support from Madeline Udine or other people in the community that actually are working on these things. So we will also have some webinars that will be not in this series, but on the side we'll advertise, given by companies like Praxis and possibly Neurocrine on their particular small molecule drugs that they're developing and how they work, what are their prospects for helping us. So that's sort of my intro. And how do we handle questions? I want to be able to handle questions at any time. So we have the chat. Do we have a question and answer, or do we just have the chat?
We just have the chat today. And so Michael, are you going to be talking, you're going to be doing the basic research update today, right? Because the inheritance is the April 1st, right?
Yeah, but this is there's always
your intro.
There's always going to be some overlap.
Okay, got it.
So,
yeah, so if people want, they can throw questions into the chat. I'll be monitoring it, or if I miss it and I don't see it, feel free to like do the raise your hand feature, or you know, if it's something to clarify about what Michael's talking about that at that moment. Otherwise, we can also do a lot of chat at the end to really process stuff and dig deeper.
I'd like to just have an open and just informal, just ask questions anytime.
All right, so pop in.
So to be honest, I've only thrown these slides together in the last few hours. So I may not have thought of things. They might not be in the best order, but it's just going to try and give you the very most basics. It's going to be a genetics 101 kind of thing. And hopefully this will improve everyone's understanding of what's going on with SCN8A and the questions that we have about when we see a genetic report, when we see a mutation, what does it mean? I'm trying to be very general here, but I'd like to get into more specifics as needed and according to your questions. During our regular network meetings, with the different ways we've split them up, we do get into specifics on particular mutations and particular families and what's going on. And today I'm just going to try and be more general about it. So the first thing I want to just talk about is, you know, what is DNA? What is a genome? What is a gene? What's a mutation? And what are the various ways mutations can be inherited? Because next week we're going to talk about, we're going to have a very specific family meeting with people who have inherited variants or mosaic situations. And I'll re-explain it then, but and then we'll get into the individual stories. Today, I'm just going to give you a background in theory, what all that stuff means. So this slide is just the big picture. So it's showing a cell in a body, and in the cell, you've got a nucleus and a cytoplasm, and the nucleus contains chromosomes, which are blown up here, and the chromosomes are made up of strands of DNA. The DNA is made up of molecules called nucleotides. There's other names for nucleotides. Sometimes they're called bases. There are 4 nucleotides or 4 bases, and sometimes we abbreviate them AGCT, which stands for, I'll show you on a different slide what that stands for. What is a gene? A gene is a segment of this double helix DNA strand that is entwined on the chromosomes. There's thousands and thousands. There's 23,000 different genes on the 23 chromosomes that we have. I'll show you that in the next slide as well. So again, just another way of looking at it, you've got the nucleus, you've got the DNA strands, and then you've got the nucleotides that make up the individual units on the DNA strand. Sometimes we could call them like pearls on a necklace. Each pearl is a different nucleotide, and there are 4 different ones. So we inherit 1 copy of each chromosome pair from mom and dad. So this is a cytochrome, this is a chromosomal spread as seen in a microscope of somebody's, a complement of chromosomes. The first 22 are called autosomes, and the last pair is called the sex chromosomes. So you've got an X and a Y. This is a male. Females have 2 X's, males have an X and a Y. And Chromosome 12 is very special to us because that's where SCN8A is. It's a gene along chromosome 12. Sometimes when you guys get genetic testing, they'll do something called a chromosomal microarray, and they'll actually look at the level above the gene to see if there are any kinds of rearrangements on these chromosomes. Sometimes there are little chunks of the chromosome that are missing. Sometimes they're duplicated. Sometimes they're rearranged. And all of those things are detectable by some process called microarray. And your genetic testing process should include microarray, because we know that some of our families do have these type of chromosomal alterations. So there are 6 billion nucleotides in the complement, 23 chromosomes. So each of these, this is mom's complement, this is dad's, eat mom, dad, each one of these coming from mom and dad. The way that these chromosomes are combined to form are what is called diploid complement of chromosomes. That means mom and dad, there's two chromosomes of each type, 2 number 1s, 2 number 2s, etc. Is a process called meiosis, which generates sperms and eggs. Eggs and sperms only have half this number of chromosomes. And when they combine, they reform the complement of 46 chromosomes, if you will, and 6 billion nucleotides. Any questions on that? Okay. Now let's just look for a second further what a gene is. So again, here's the idea that, you know, you've got these chromosomes inside the nuclei. Then the DNA is makes up the chromosomes, and the nucleotides make up the DNA. And a gene, and here are the 4 nucleotides, and that's why they're called A, C, T, and G: adenine, cytosine, thymine, and guanine. When you get your genetic report, you'll have a C dot, and then you'll have a number, and then you'll have a C to A, or C to T, or G to A. That little caret sign, that means one of these has been substituted for another, and that's what a mutation is, and we'll talk about that now. But before we get to that, let's talk about what a gene is. So, you know, a crude definition of a gene is just a segment along the DNA on a chromosome. But what makes up the segment? Well, it's got a very specific set of instructions that, well, let me just talk about the process of replication. Is when cells divide, all of the chromosomes have to be reproduced every generation. So that's called DNA replication. But when a gene is a unit in which there's something called transcription, which means an RNA molecule is made off this based on the sequence of the DNA. So the DNA holds the instructions to make RNA. RNA is transcribed from a section of the chromosomal DNA, and there are instructions for telling the molecules that make RNA where to begin and where to end. And so there's a transcriptional process. Interestingly, there are pieces within the gene segment that's being transcribed that are spliced out. Like when you make a movie, you splice certain things out, and then other frames are connected in a process called splicing. Well, genes in humans are complex, and one of the levels at which they're regulated is by cutting these pieces out. Now, we've heard about splicing mutations. Some of our families have splicing mutations where this process is not occurring typically. But most families have other types of mutations. We'll talk about it in a moment. But this just points to the idea that splicing is a process, and it can go wrong, and it can lead to a misshapen protein. So when the final RNA is spliced and everything's put together correctly, then it's translated. So the third process is called translation, and that's what makes a protein. So you could say a gene is a DNA segment that has the instructions for making a protein. There are other kinds of genes too that do not make proteins. They're sometimes called regulatory genes. So genes that will affect the expression of other genes. So it's a very complex engineered process. We don't understand it fully, but the basics are clear and they've been known for decades. And how disease fits into this picture is what we'll start to talk about next. But the main idea is, in a particular case of a protein, it folds in a three-dimensional structure. We're looking at it two-dimensionally here, but it will fold into a structure, and it's usually the structure of the protein that really is the critical thing, that when it's altered, it can affect the way the gene functions or the protein functions. So sometimes we'll talk about the gene that's malfunctioning, but really what we really mean is that the product of the gene, as it's transcribed and translated, it's the final product that's misfolded or not functioning properly. So in the case of SCN8A, we know the gene is SCN8A and the protein is NAV1.6. All right, so why do things go wrong? So we're all familiar with the idea of a mutation. So a genetic mutation is a change in your sequence that happens during cell division, when your cells make copies of themselves. So like I said, every time a cell divides, and that cell division is a critical process in our body. We start out as an embryo. Mom and dad's sperm and egg meet, they form a zygote. The zygote develops into an embryo. The embryo's in the process of cell division, and it forms two cells, form four form eight, 8 times 16, and on until you're into the trillions. And that's our body. It's made up of trillions of cells that all come from the process of cell division. Well, every time a cell divides, the entire complement of our genome, the 6 billion nucleotides, 3 billion from mom and dad, each of those nucleotides is copied and made a new chromosome from the previous one in that process called replication. And here we show a DNA strand. In order to replicate, it unwinds and then it copies itself on each side of the strand. Now, amazingly, there's 6 billion of these, and only a few mistakes are made each time a new person is born. So the error rate is such that there are maybe 50 to 100 mistakes made each time a genome divides from one parent to child. And you think about it, you say, well, 100 mistakes, that's terrible. No, it's an amazingly complete and accurate process with just a few tiny mistakes. Now, depending on where that mistake is made, it may not affect the gene. It may not affect anything, because we only have something on the order of 23,000 genes that are protein coding, but we've got lots more DNA that doesn't do anything that we know of. Some people call it junk DNA. Some people say it's important DNA in a regulatory process of some sort, or it's structural, but mutations don't affect it in a way that would cause a disease. So where this mistake happens is critical, as we know. So we know that a mistake occurred in the SCN8A gene, which is why we're here together. Here's an example of one type of mutation. It's called a point mutation. It's just one nucleotide is substituted for another. So an A in the original parent strand is a G in the child strand. And like I said, that may make a difference or it may not. There are other types of mistakes that are made. There are deletions, just like what I was saying for chromosomes. There could be a nucleotide that is deleted, one that's added, and then different types of mutations that we'll talk about that are really critical to understanding how different mutations in SCN8A lead to different phenotypes, different severity, different loss of function, gain of function. What is all of that? We will talk about it. So as you can see, we're getting a little bit more complex as we go along here. So I do want to open up for questions at any point. And I'll stop for just a few seconds so I can sip my tea.
Oh, good. Michael, that gives me a chance to ask a question. So this is Jada, and I just came off a 2-day meeting on biomarkers. And part of the discussion was the potential for using more sophisticated understanding about genetic mutations and the full genetic background as an early indication to predict, prevent, target better treatment. So my question is, so we've got this era that's occurring at conception, is it? When the actual the 23 from each meet, so there's this era occurs right then.
You're leading up to the next section of my talk.
All right, but let me still try to ask. But the age of onset of this era showing up varies quite a bit, as we know.
Yeah.
So should we talk later about that potential to by better understanding the era that we can develop, I guess it's gene therapies, but it's really targets for research of how to intervene. And I guess they've got a pilot or a program actually in tuberous sclerosis where they found a target and they're working with infants. They can immediately detect what the problem is and they start treating it.
Yeah. If you can detect these errors before the child is born, then you can be ready for it at birth. That's in vitro testing. They're very good at doing that for chromosomal anomalies, and not so great for detecting this type of point mutation. For chromosomes, you would just have to get a sample from the amniotic fluid and then make a chromosomal spread on a slide like this. And then you would see, oh my gosh, we've got trisomy 21. There's an extra chromosome 21. Then you know you have Down syndrome. But the problem is developing assays from amniotic fluid that would detect any type of mutation of the type we just mentioned, a single point mutation. That's tricky. You'd have to sequence the genome, or you'd have to have some specific assays. Let's say you had an assay to detect all the SCN8A variants that are possible, because you don't know which one it could be. And then you would have to detect everyone possible at the particular gene before the child is born. Now, in the future, if we can imagine a future where that's possible, then if a particular mutation that we're learning has a particular outcome, like a gain of function or a loss of function, which we'll cover in a minute, you might be able to treat that at birth before the first seizure. That would be amazing. So this neonatal or prenatal testing is something that people are working on. I don't know the state of the art in terms of how far they've gotten in terms of detecting point mutations like this. There may be companies that, as the last time I looked into this a few years ago, there were companies working on this, but it would have to be gene by gene by gene. So that would be a huge benefit, right? To know that you've got this and that you can be prepared for it when the child is born.
Will it be more feasible to maybe test that in some of the inherited cases where you know you've got a parent who has something? So it could, because you were describing that they would have to look at the whole genome, but if you know there's a parent or two who have something, is that basically the time and
Yes.
Yes,
so I'm going to show inheritance in a minute, and we can talk about it. Jada, if you could take some notes on the topics that we are bringing up that I have not covered, like prenatal testing.
Okay.
And anything that you have, let's write them down, and I will fill them in as we go. Next time, I will do more work to make this tailored to your questions.
Okay.
And Matt had his hand up. Matt, did you want to ask something, or?
I was just going to ask, so when, Dr. Hammer, when you're saying an error, can that be interchanged with like the de novo when we say that this is something that's de novo? Or is that what you mean when you're saying an error is like, this is just a random occurrence? I
Yes.
feel like I've heard that term a lot. And so is that
Yeah.
what is that what we're saying right now?
Yes, there are. This is the first time the mutation is occurring, and it's de novo when it's the first time it's occurred. If it's inherited, so we're going to talk about inheritance after I talk about mutation, and we'll get to that very specifically. I think save your question for a few minutes, Matt, because we're probably going to cover that.
Okay, cool.
Okay, so when do genetic mutations happen? They occur during cell division, as I said in the last slide. And I wanted to make you aware of there are two types of cell division. And again, it's the process by which all the chromosomes are copying themselves where these mistakes are made.
I think he's gonna get by it soon. I think he's doing
Jay, I think you're not muted yet.
right. You're not gonna change him now.
Kabi, I think you better mute Jayetta. She's on the phone.
Jayetta, can you mute?
Yeah, sorry.
So mitosis is the process of making new cells for your body. Like I said, when your cells are dividing from the very time you were conceived, that's mitotic cell division. And during mitosis, your genes instruct your cells to split into two by making a copy of your chromosome. So again, that's just mitotic division. Parent cell, DNA replicates, and then the cell split. So the replication occurs before the split. The spindles are these little fibers that latch onto the chromosomes and pull them apart and make a complement of 23 chromosomes, 46 chromosomes in each cell, that each daughter cell. If there's a mistake made there, it's a somatic mutation. We'll talk about somatic mutation in a minute. Meiosis is the specific process of making an egg and a sperm. So before the parent cell is produced to go into its specific meiotic process, there's mitotic processes that precede it. And we'll talk about that in a minute as well. But in this case, the two daughter cells only have half the number of chromosomes, 23 each, so that when they meet up in the zygote, you get back to the 46. So during meiosis, chromosomes copy themselves with half the amount of chromosomes as the original, and that's how you're able to get your genetic material equally from each parent. So that's a very sort of nutshell things about mitosis and meiosis. When we talk about somatic mutation in a minute, we'll talk about mutations that occur here. And when we talk about germline mutations and the ones that are inherited from mom or dad, we'll talk about this process, meiosis. So that's what's shown here. So germline of the sex cells that we just talked about are in the meiotic process. If there's a mutation, all the zygote, all the cells of the zygote inherit it because it's in the sperm or the egg. So it's right from the get-go. The individual gets only one mutation in one gene, one copy of the gene. So it's occurring on dad's chromosome, not mom's, or mom's chromosome, not dad's. So the zygote has all the cells have that mutation, but remember, it's only got one copy of the mutation and one wild type copy. Wild type means the original copy that wasn't mutated. So the entire organism carries that mutation, but when that individual undergoes meiosis, because it's segregating chromosomes out again, only half those gametes get the mutation. So think about that for a second. Does it make sense? The entire organism has the mutation, but the gametes, only half the gametes have it because it's heterozygous. It's in one copy. So when you get your genetic report, it will say autosomal dominant heterozygous, means it's on one of the autosomes. One copy of the mutation is enough to cause the phenotype or the disease. And you have one mutated copy, one original copy. And if you have children and you have that mutation, and it's a de novo mutation that occurred in the sperm or the egg of your parent, you have a 50% chance of passing that on. That's the key process we need to understand. If the mutation occurs later, it can occur later. It doesn't have to be the sperm or the egg. It could be in the zygote itself. during a mitotic division, then there'll be a little patch of cells that have the mutation. If it's in the shoulder area, it's not in the germline, and so none of the children will get it. But some part of your body has it. So who knows if some of us don't have an SCN8A mutation that occurred in our zygote that's doesn't affect our brain. If it occurred in the cells that lead to the brain, then it's possible that that individual will have epilepsy, but not pass it on. And I don't know any cases like that. We wouldn't know because that would be tough to find out. So the timing of the mutation in terms of this whole process of cell division and zygotic formation and development into an individual is critical to us thinking about how we may pass on something or how we inherited something. So now we're going to go to the more complex question of mosaicism. This individual is a mosaic. Okay, Matt, your question is here. An inherited mutation. A father has the mutation in all of his cells and he transmits it to the child. That's this situation here. It's inherited. It's not de novo with respect to the child. It may have been de novo with respect to the parent. Then here's a case where the father has a mosaic mutation that affects his germline. So instead of the shoulder being affected, and it doesn't have anything to do with his brain, his gametes are affected. And he doesn't have any brain issue. He doesn't have SCN8A affecting his brain, but he has a mutation in his germline, and it gets inherited by his child. So that's a form of de novo mutation with respect to the child, and yet it was occurring in the father's germline somatically. That's actually called a gonosomal mutation, gonosomal. A parental germline mutation is a father has a germline mosaic mutation, transmits it to the child. So in this case, one of his sperm was affected by a mutation, or a small number of cells in his germline were affected, and he passes that on to his child. But not all of his children will get it. And then finally, the de novo is he has a mutation in a single sperm and it transmits it to the child. So not his germline. He doesn't have that sperm anymore. It was just a single sperm. It was the one that fertilized the egg, and the child got it. That's a de novo mutation. This one is, oops, he's still got sperm that have that mutation. Not all of his sperm got it because it was a somatic mutation in his germline. So we don't know how often he'll pass that on. That's the scary thing. That's the one where, oh my God, I might be a germline mosaic. I don't know what percentage of my sperm that I'm still got in my body will have that mutation. It depends on how early in my development that mutation occurred. Just like, in this case, if that mutation occurred very early on, maybe this person would have patches all over his body versus a single patch. Well, if it's in the germline, we don't know how big that patch is in the germline. So de novo, most of us are de novo. Either the sperm or the egg had a mutation, and some of us inherit it from a mosaic parent, and sometimes the mosaicism in the parent is not in the germline, and sometimes it is. And sometimes the mutation is an entire body of our parent because they inherit it from their parent, and they inherit it from their parent. In fact, here's what a pedigree looks like in where you have an inherited mutation. And I'm going to use this slide in a couple of different ways. So here's great-grandparents, grandparents, parents, and child. So here is, we don't know where this person got the mutation. Maybe it's de novo, we don't know. But this woman, the woman is indicated by a circle, the man is a square, passed this on to 1, 2, 3 children. a boy and 2 girls, and they each married somebody who's not in the family. So it's fresh new DNA is getting in the family. This variant is inherited by all 3 children from this woman, and then each one of them passes it on. So here's a whole bunch of kids. 1, 2, 3, 4 of the 8 inherited it. 4 did not. 1, 2, 3, 4. So the dot shows an inheritance, and the shading shows how severe the effect is. And here's a family, another family where 4 out of 6 inherited it. Here's one where 3 out of 5 inherited it. And these symbols are just different forms of the epilepsy, febrile seizures or other types of seizures that occurred, but they were all mildly affected, so such that, you know, they went on, had a normal life, they had kids. So in order for something to be inherited, you have to reproduce. You have to have a mild mutation that doesn't affect your ability to find a mate, get married, have a child. If the mutation is severe and the individual is affected in a way that will prevent that person from either growing up to get to the age of reproduction or having the capability of marrying and reproducing, that variant will not be inherited. It'll be just in that one person. If it's this case, then it just goes passed on from generation to generation. So some of us have that situation where we have an inherited variant. It implies that it's mild, but there's another way around that. In this case, there are mildly affected individuals, there are unaffected individuals, and there's one individual who has DEE. And so this particular situation is super interesting because, like I said before, there may be other genes in the genome of this individual that may have come from outside the family or from a new mutation at another gene that made this more severe in this individual. So that's called the genetic background. And we all have a different genetic background because when chromosomes are dividing in this process of meiosis, we each get a random set of chromosomes from mom and dad. We all get a different complement of chromosomes. If you're brother and sister, you're 50% related on average, but you may have different chromosomes from mom and dad because it's like shuffling a deck. I've got a deck of 46 cards. I'm going to shuffle it. I'm going to put 23 in this pile, 23 in this pile, and then I'm going to merge it with mom's shuffled deck, make 46 again. And we all have a different complement. So we're all different. We're all unique. And then you add mutation on top of that, that makes us even more unique. But the point is, if you have an inherited variant and it's the same variant segregating in the family, each one of us has got a different background on which that variant is expressed. And sometimes there are modifiers that make it more severe or less severe. So we always have this discussion. Why I have the same variant as person X, but we express it, we are different. We seem to have different effects. That could be genetic. It could be, no, it's the same SCN8A variant, but we have a different genetic background. So we're modifying that variant in its expression in our body differently. Or it could be environmental. It could be, I grew up in one set of environment, you grew up in another, and that affected us. Or I was genetically tested at 2 days after I had my first seizure, and you were genetically tested 10 years after you had your first seizure. There are all kinds of things that could lead to different expression of the same mutation. Now, we're not talking about an inherited case there. We're just saying people with the same de novo variant, which happens occasionally, and we'll talk about that as well. But in a family, next week when we have our inherited families getting together, we will find out how different that gene that they inherited, the same gene variant, is affecting different family members. And that's a controlled sort of experiment in a sense that the family members probably have a very similar environmental background. They inherit the same variant, but the variant is now being expressed slightly different in different individuals in a way very much similar to this family here. So let's open that up for any questions, because I know it's quite complicated. These are questions that individual scientists might devote an entire career to studying. I was very interested in modifier genes for a number of years, and this is actually a family that I studied, and we sequenced the genome of the exomes of this entire group over here. Now, this was back in the day when I had funding from the Dravet Syndrome Foundation, and what we ended up doing was finding a variant that explained why the 3 individuals here didn't have epilepsy at all. They were normal, typical, no seizures, nothing. And we found out why that was the case by finding there was another mutation that those individuals inherited from this woman here that protected them. So they were protected. That happens in SCN8A as well. We just don't know very many examples. It would be really cool to find out what protected those individuals from having epilepsy, because if we could reproduce that with a drug or some kind of treatment, we would have the genetics teach us how to do that. That was the purpose of looking at modifier genes. Can nature tell us what the secrets are to preventing this disease?
So we did have a question from Joanna that came in saying that she has a child with the same mutation as herself, but a second child that does not. And she's asking if the second child is still going to pass on the SCN8A if she doesn't not have the mutation.
So in the first one, the most likely scenario is that, like I said, she passed on. Which slide was it? Here. So she has the mutation, and one child, now she's a female, so these would be eggs, not sperm, obviously. But half the gametes have the mutation and the other half do not. So I would say the most likely explanation is she passed on an egg, was fertilized that in one child, and another egg for the other child, and one egg had the mutation and the other didn't. Just by those are Mendel's rules, Mendelian genetics. The other possibility, though, is if it is a mosaic, if Joanna's a mosaic, it's possible she could pass it on. Oh, would her child pass it on? So if her child, it gets complicated, right? Because maybe that child is protected and has the mutation and passes it on to somebody who's not protected. That would be another explanation. I think that's less likely than just the Mendelian situation, where it's just cost coin, 50% of your kids get it and 50% do not. Does that make sense? It's possible it's more complicated. It could be that your 2 children are here and one has it and expresses it and one has it and doesn't, and then this person will pass it on. That's less likely, but possible.
Yeah. Steve, do you want to ask your question?
So, Mike, are you saying that the de novo is coming from a mutated zygote? Is that, or is it coming from a mutated sperm or egg?
Mutated sperm or egg.
Okay.
So you see here, and sorry, this is a little bit small, but in this particular case, the dad had a de novo mutation leading to a mutation in a single sperm, and that was passed on. That's called the de novo situation.
Okay, so it's not internal within the reproductions?
You know, it's like the germline is like a separate lineage than your somatic lineage. It's not in his body in a sense, because it was just in his sperm. There's some fascinating experiments. People figured out the germline by these guys would cut off the tail of a mouse and they would see if the offspring inherited the cut off tail. This is way back in the time, right? And they would do it over and over again. And they found, oh my God, the cut off tail never gets inherited because it's a somatic change. It's not in the germline. So they figured, oh, there must be a different package of cells that's not in our body necessarily, but we pass on. And so the germline was discovered. And So in this case, the de novo mutation is affecting the germline only.
Okay, so I was just a little confused because I thought de novo was basically unknown as to where it came from. So,
Well, it's random. It occurred randomly one time in that one sperm. So it was in a process of cell division leading to that one sperm. It was probably in the process of meiosis, not mitosis. If it occurred in mitosis leading to the sperm, then there might be other sperm that individual still carries in their body that have the mutation. But if it happens in meiosis, then it's only happening in one product of meiosis, and that sperm is then given away.
okay, thanks.
It's complicated, I get it. It's sort of like, it's the same process of mutation, but depending on where it hits in the process of the body developing, tells you what's going to happen next. So imagine an assembly line, and you take a hammer and you hit the product on the assembly line. If you hit it early on, it might affect a lot of downstream things, and if you hit it late on, it's just going to affect that one thing. So that's not a very good analogy, but you get the idea. Because it's a hierarchical division process. And if it happens early, everybody gets it. If it happens late, just one gets it.
And Zipporah was asking if you can have somatic de novo. Is that?
Yes. So this person has a somatic de novo. It doesn't affect their brain, but if you did have a somatic mutation, so the body is made up of stem cells. The stem cells are not particularly like a liver, brain, or heart cell or a germ cell. And those stem cells give rise to specific differentiated cell types in the body. When the conception occurs, the first many cell divisions are stem cells that eventually differentiate into other cell types that become your eyes, your brain, your liver. Again, if the somatic mutation occurs early in the zygote, then your brain, your heart, and your liver will all get the same mutation. If it occurs much later, after the child is formed to some degree, then your heart might just get it, or some patch of skin. And there are cases of people who actually have mutations that change the color of their skin, and you'll get patchy skin because this patch was derived from the stem cell that got the mutation, and this patch was not. And so I could go find some pictures online of somatic mutations that affect people that make them look patchy. Or if you think of a calico cat, the patchwork of colors on a calico cat are due to a mutation that occurred in a cell that gave rise. So black and what do we call it when a cat's orange? Orange and black are like two different alleles of the same gene. And a female cat will have two X's, and you'll see that the X that has the black gene will make the black patch, and the X that has the orange gene will make the orange patch. And that's why females are usually calico and males are not, because they only have one X. That's that's another form of mosaic. So anyway, I've probably gone beyond what your question was. I hope I answered it, Zipporah.
You did, thank you.
And I know, Megan, you got your hand raised. Did you want to ask something?
Yeah, so I guess kind of what you were saying about a gene protecting, for us, I guess it would be the opposite with Chloe, where a gene has possibly magnified the SCN8 that's inherited from father.
You think Chloe is so Chloe inherited something and she's more extreme than her dad.
Yeah, he, as far as we know, doesn't really have any symptoms of it, but she has that duplication on her microarray.
So dad has the duplication?
Dad has the SCN8A.
Okay, he has
but Chloe has a microarray duplication on XQ28.
And she has the SCN8A.
and she has the SCN8A.
Yeah. So who has the duplication on the X? Anybody else in the family?
It's more than likely me because her and Peyton both have it.
Okay, so let's look right here at this little piece of the family tree. Here's you. You've got a duplication on your X. Both your kids got it. Here's your husband. He's got SCN8A. Chloe got that, plus she got the duplication. So maybe Chloe's more severe because she's got the combination of the two. Nobody else in the four of you has the combination of the two. That would be a possible scenario, right?
Yes.
Okay. That's what I think's going on. But, that's just a hypothesis.
Yes.
Okay. I know this is complicated stuff.
I think just one thing that I just wanted to go back to, Dr. Hammer, was so it came from somewhere within whether it was the sperm, the germline, or the pass on from the actual genetic from the parent. Before this conversation, my understanding was, or the way that it was explained to me was like, if both parents don't, because Liz and I both got tested for SCN8A, neither one of us had that. And so it either was, so when we say de novo, it was either the sperm or the egg had that.
Yes.
Potentially. Got it. Okay.
Yeah.
I always thought, or I just thought it, or I guess the way it was explained to me before was it just happened while those cells were being put together, but obviously it was somewhere within that process. So now that helps. So I appreciate that. Thank you.
Yeah, I mean, the most likely scenario is that it happened during meiosis, the process of producing your gametes, your sperm or your egg. And if it happened there, then it's done. You don't have it in your body anymore if it's just in one. But it's possible that it happened a little bit earlier in the process of making your gametes, and so that you may still have it in your body, but you wouldn't know it because it's not affecting your brain. It's just affecting your gametes. And that's less likely, doesn't happen as often.
And is there testing that you know of that will
next week we're going to talk to families where the daughter has a mutation and the dad is a mosaic, and the daughter has it in her entire body, and the dad has it in his blood. He has it in his germline. They didn't test his germline, but they tested his blood. So if your blood cells get the mutation and you give a blood sample and you have the mutation too, but you don't have any effects of the mutation, chances are you're a mosaic. It's in your blood cells because that's where they found it when they did the test, and it's in your gametes because it was passed on to your child. So if we find the same mutation in your blood sample that is in your child, that's how we discover the mosaic. It could be that it's not in your blood cells, it's in another part of your body. And so it would not look like a mosaic. So there are false negatives on mosaics. The fact that we test somebody and find out they're mosaic means we were lucky enough to find it in the sample we took. And we're not going to take a piece of skin or a piece of heart or your gametes. We're only going to take your blood sample or your saliva sample to make a genetic test. So that's the complicated thing about discovering somatic mosaics. So, yeah. And then some people can be a mosaic for SCN8A and have epilepsy, but be milder than those that have it in all of their cells for obvious reasons. So maybe they only have it in a certain percentage of their brain cells. And so they might have a milder effect. There are cases of that in our community as well. So just some details on your genetic report. So we've covered this kind of stuff before, but I just want to make sure everyone understands that because we have this process of transcription and translation, we go from DNA strands to RNA strands to proteins. And the way that works is every three, so there's 4 different possible nucleotides, A, G, C, and T. And they're random, they're kind of distributed on the DNA, and then it's copied into RNA. RNA uses a different form. It's called uracil. So T's are actually uracil. So don't get confused by that. The point I want to make is that every 3 nucleotides makes a single amino acid. That's the genetic code. And that's Gabi's design, her decoding developmental epilepsies design is based on this process, where a methionine, a proline, a serine, a valine, they are amino acids. There's 20 essential amino acids that correspond to these triplets. And there are different sequences of these triplets that can mutate and not lead to an amino acid change, and other ones that can. But that's, I don't want to get into too much detail there. But this is the process by which DNA goes to RNA, goes to protein. And the reason that's important is if there's a mutation in the DNA and it leads to a particular mutation in the RNA that in the triplet form leads to a substitution of an amino acid, then you have a substituted amino acid. There are cases where the mutation in the DNA leads to a change in the RNA that doesn't lead to a change in the protein. We don't worry about those. They happen all the time as well. So that's what happens. That's how we get an amino acid change, a particular mutation in the DNA, which leads to an RNA change, which leads to a protein change. Now, the proteins, there's 2 different codes for proteins. You've seen this in your genetic reports. There's a 3-letter and a 1-letter code. Here are the 20 essential amino acids. Aspartic acid is ASP in the 3-letter code or D in the 1-letter code. Threonine is, so you don't have enough letters that begin, in other words, there is an A somewhere here. It's alanine won the day because it got the A, but aspartic acid got the D. There's other cases like that where, like G for glycine, but unfortunately glutamic acid, that G was already taken, so it had to get an E. And that's why some amino acids, here's glutamine's another G, but it got a Q. So the amino acid name, the first letter doesn't always correspond to the 1-letter code, but sometimes it does. There are more than one T, so we got a W for tryptophan. Threonine won the day and got the T. So that's the 3-letter, 1-letter code thing. So when you get your mutation, you'll see the first 3 letters will be the original amino acid. The number will be which amino acid in the chain, and then the next 3 letters will be the substituted amino acid, or it'll be a single letter code change. So when you look at the C, it's the change at the DNA level. And if you look at the P, it's the change at the protein level. So here's an example where in the normal, before the mutation occurs, you have this string of nucleotides, in the RNA, and this leads to, these 9 nucleotides lead to 3 amino acids, ARG, HIS, and ALA. This would be R, this would be H, and this would be A. The number that mutates in the DNA is given the C dot. That's right here. And then we got an A for a G, which in the we got an A for a G, so that means, so here's the original G, here's the mutated A, in the RNA it becomes a U, that's irrelevant, and then it's going to substitute histidine for tyrosine. So it goes RYA, and so you've got the position in the protein that's 5, 6, 7, and it went from H to Y because histidine is H and tyrosine is Y. Does that make sense? It's really all about the genetic code, but the C dot's telling you the DNA level change, and the P dot's telling you the protein level change. If we look at our gene, SCN8A, in its protein form, all the changes in the amino acids that we know of are shown here. And there's 1,980 of them starting here. Here's number 1, and here's number 1980. So there's 1,980 amino acids in this chain, or this necklace of pearls. In this diagram, each amino acid is shown as a little open circle. If it's colored with a little light blue pattern, that means there's a variant in an individual at that site, in an individual that doesn't have any neurological disorder. So we assume that the blue dots are mutations that occurred and are inherited for many generations, depending on when they occurred back in time. So the population history is another level of the way the body develops. The body develops from a conception leading to a zygote, and then the process of hierarchical differentiation of those cells to form the individual. The earlier the mutation, the more of the cells that get it. Well, the earlier the mutation in time in a population, the more individuals that inherit it. So those are called polymorphisms. And sometimes, things are at 50% in the population because they occurred a long, long time ago. Or they're in a specific ethnic group and they occurred recently in time, maybe 30 generations ago. And so only certain people of a certain shared history have that variant. It's still a polymorphism. It's just not shared. It's only like 2% in the population as a whole. So these blue dots are polymorphisms that range in frequency from 1% to 30 or 40%. And that's how variation distributes in the population, in the human population. And sometimes those variants are favored by evolution, so they increase in frequency. Sometimes they're disfavored, so they are selected out, but it takes time. It takes many generations to remove those deleterious variants or to increase the frequency in an adaptive sense. So that's population genetics. The variants that we're talking about are created the very same way the variants in SCN8A are created, but the variants in SCN8A are different in that they are serious. They cause seizures or epilepsy or autism. So then suddenly we're in a different framework. We're now into a monogenic disease that is unlikely to be at high frequency in the population because most of the individuals don't survive and reproduce, or survive to reproduction or reproduce. So the stars are going to show you the variants that are found only in our population of patients. And if it's a yellow star, it's just found in one individual. And if it's a gold star, it's found in multiple individuals. And we have some that are found in, this is old, it's not up to date, that are found in as high as 50 individuals. The same site is mutated. And so those variants I'll talk about in a minute are very important because now we can study individuals who have the same variant to understand if that variant is predictive of the severity of the outcome, the clinical severity. We don't know. Up until now, people haven't found that to be the case, but we know it to be the case. We are using registry data to prove that to be the case. And as we speak, we have a manuscript that is based on the registry data that will be submitted to Epilepsia in the next few days that shows that certain variants have very common outcomes, that enough that we can statistically identify and use that variant as a predictor of outcome. I'm not going to talk about that today, but
Michael?
yeah.
Could you go back one slide?
Yeah.
I wanted to better understand the difference between the C dot and the P dot. So you said the P dot is the protein and the C dot is the RNA?
DNA.
DNA. can you, so you always focus in on the P dot.
Yeah.
And that's the numbers that are in that protein that you always show us. What is the C dot and what significance is it? And
Well, it has a different numbering system because it's, I don't have a picture of, let's see if we go to, the gene, going back to the original slide of what the gene is. So the C dot would apply to the numbering system in the gene, but remember, because of transcription, translation, the amino acid number is going to be, you would divide the gene number by 3 to get the protein number. Because it takes 3 bases in the DNA, 3 nucleotides, that encode a single amino acid. So I'm going to count my amino acids 1, 2, 3, but that's going to be 3 amino acids is going to correspond to 9 nucleotides. There was a slide that showed that, right? Here. So if I'm counting my cDNAs, I'm going 1, 2, 3, 4, 5, 6, 7, 8, 9. If at the protein level, I just 1, 2, and 3. So it's confusing. I like the protein numbers because I can correspond those numbers directly to this. And I can say, oh, 850 is in D2S4. And so 850 is the number I pay attention to. I don't care so much about the cDNA number for that. I don't even know what it is off the top of my head. It's going to be 2000 something, something, something. It's going to be 3 times 850. So I just pay attention to the protein number. Does that make sense?
So it's not that it's something different. It's just its place in the gene, which has a different numbering system. So it's not 2 different identifiers of what your mutation is. It's 2 different ways it's counted in the gene or in the protein.
Exactly.
Okay, and the protein that you show us all the time is the more useful way to understand variants in this particular gene.
Yes, exactly.
Okay, that's helpful.
Now, there's one exception to that, and that is sometimes the change in the DNA doesn't lead, it's a change in the DNA in a place that doesn't cause an amino acid change, but it causes a splicing change. So in that case, we're not going to see an amino acid change result. So some of you on your genetic reports will have a C dot and you won't have a P dot. And then we go, oh, it's C dot minus 2 bases. So if there's a small deletion in a splicing area, the coding for the instructions for splicing correctly have been messed up. So then we go and look at the C dot and we say, oh, that's a deletion or an addition in an important region that would affect splicing. So that's an exception. That's the only exception. There are more complicated mutation types as well, but they're all related to that whole process of splicing.
Teresa just had a question in the chat.
Okay.
Do you want to ask it yourself, Teresa?
Yes, sure. I'm sorry. Does that mean that that's when a gene mutation is now considered a variant of unknown significance?
Sometimes there's 2 ways that you can get a variant of uncertain significance or unknown significance, and that is, it could be that it's in the DNA level, there's no P dot, and we don't quite know what the effect is. Or it could be that we do have a P dot, but the mutation is in a region of the gene that has a blue dot, or a region of the gene that just doesn't have many yellow dots, yellow stars. And so then people are going to go, well, nobody else has that variant, or somebody else has that variant and they don't have epilepsy. So then they go, well, it's not in a region of the gene that we understand, like these loops are more difficult to understand. So then they'll say it's uncertain. the way they might decide that it's pathogenic, in other words, not uncertain, is that somebody else has the same variant in their database and they have epilepsy. Then they'll go, okay. The other way is they'll say, well, is it inherited? If it's de novo, that also supports the idea that it probably is the causative variant. If it's inherited, the person making the call on your genetic report might go, well, it's inherited from someone who doesn't have epilepsy. I'm going to call it uncertain. So they try and standardize the way that people who call these variants as pathogenic or uncertain do it in the exact same way. But they've done studies on comparing different laboratories, and they're not perfectly consistent. The guidelines for calling these things still have room for different people making a different decision. And that's problematic. But they really try, they have guidelines called the ACMG guidelines that are supposed to tell people how to do this and try and make it very strict. But the way, let's assuming they do it perfectly well across different laboratories, then there's still going to be cases of uncertain significance that arise because the variant is in a region that's not understood, or it's not shared with anyone else, or it is shared with someone who doesn't have epilepsy. And then I'm not so quick to dismiss it because there are these more complicated things that could be going on, like the protective modifiers or the genetic background, or an interaction between SCN8A and something else. Again, a modifier genes. Modifier genes are not well understood, so you can't discount something as being not pathogenic just because it was inherited from someone without epilepsy, or there's somebody else that has it who doesn't have epilepsy. So I like to think of it more deeply than that.
Can I ask a qualifying question? So people talk about 8A as a monogenic disorder, but a lot of families have VUSs in other genes. Is that maybe we're not as monogenic as people think, that or it's just that it's monogenic and there are all these modifiers, good and bad?
Yes, the latter. So
Okay.
it's monogenic because SCN8A, mutations in SCN8A are strong enough to have an effect on their own. And in most people, probably it is the only thing affecting the patient, the phenotype of the patient. But then there's a spectrum of the disorder that ranges from mild to severe. And some of that might be due to other genes playing a role in those individuals. Some of it might be due to the fact that each variant has a stronger, milder effect. But it's still monogenic no matter what, because SCN8A has got a strong effect on our phenotype, is just the overall, saying the combination of symptoms that we have. But yes, it is monogenic. There are lots of complex diseases. That means that there are hundreds or dozens of genes that contribute to them, things like diabetes, obesity, well, even non-diseases like height, how tall you are, is lots of genes, or your eye color. There's a handful of genes that contribute to eye color or skin color, a handful. Diseases, there are diseases that are due to the smaller effects of multiple genes. But when you have a gene with large effect size like SCN8A, it's a monogenic disorder, not that it couldn't be modified by other genes.
Thanks, Michael.
Steve, did you have your hand up?
Yeah, I did. The question I have is, pedo came through as likely pathogenic. Is that because he was the first, or is it his?
Yeah, so there's a 5-level scale for the way these laboratories call variants. There's pathogenic, likely pathogenic, variant of uncertain significance, likely benign or benign. So it's just 2 levels of uncertainty, essentially, if it's pathogenic, likely or pathogenic. If it's likely pathogenic, it's probably pathogenic. If it's pathogenic, it's slam dunk pathogenic. So he came up as slightly under slam dunk, right? Because they didn't know that 1309, it wasn't found in anyone else. The rules would say if it's not in anyone else, but the person has a phenotype or symptoms that go with other people that have variants in the same gene, even if it's not the same one, then they might, and it's in a region of the gene that makes sense, like it's in the transmembrane region. So 1309 would be somewhere down in here, I think. I'd have to go look for sure, but it's in this transmembrane region where you can see, if you squint your eyes, you can see most of the mutations that affect epilepsy are in this transmembrane region. Some of them are in loops and linkers and things too, but they would say, oh, they research the gene, they research the number of people that have it or don't, and then they make a decision. And so I guess in Peto's case, they were likely pathogenic. And so now we know it's, the more we know about Peto and the more we know about Peto's variant, the more we know it's definitely pathogenic. It's just we don't quite know what its effects are. And Peto is, you might share that. Do you want to say anything about Peto's variant that you just learned about?
Oh, yeah.
Peto is neither loss or gain of function. Peto, they're very confused, but they're thinking that it is making his protein act like it's an SCN, it's a 2A, 5A, or 9A. So that's what we know. And yeah, just add more to the confusion. So, yep.
Right. All right. We'll get to gain of function and loss of function in a minute. I'm not going to go on for too much longer because I know your brains are getting full. But I wanted to talk about how this affects us in a more practical way. So let me see. Maybe I'll pick a few of these. So, you know, we know that mutations in ion channel genes, you know, change the way these important channels function that play a role in regulating neuronal excitability. Okay, so that's a known. SCN8A encodes a protein that sits at a very important part of the neuron, and it really affects how much nerve conduction is going to go on, or action potentials are going to be fired. Action potentials are the electrical energy that the neurons fire to communicate with other neurons, and the rate at which they do that and how often they do that it leads to excitability. Excitability is good. You know, you need excitability to have things happen, but there's a balance between how much excitability and how little excitability is going on. So, you know, when we talk about gain of function or loss of function, a gain of function is a mutation affecting the channel that lets too much sodium in, either because the channel opens prematurely or closes late. It delays inactivation. That generally leads to seizures. And then loss is when it's quite the opposite. There's too little sodium getting in because the channel's not opening properly, or it opens late, closes early. There's various ways it could not let enough sodium in, and that generally leads to developmental delays without seizures or with a different seizure type. And we'll talk about that right now. So the types of mutations that we see, like we call it a missense, just like we were talking about before. And aspartic acid, you know, so you have an asparagine to aspartic acid at position 1768. That's in this case, it happened to be de novo, but it was a change at 1768 from N to D. That's a missense. A nonsense is when you change a nucleotide that alters the way the protein is transcribed, such that it stops transcription where it should normally allow the transcription to continue, but it puts in a stop codon. So one of the essential amino acids is not an amino acid at all. It's a 3-letter code that says stop transcribing. And if that happens in a wrong place, then the protein chain never gets produced. So that's a loss. It's called a nonsense. A frameshift is when a occurs because perhaps an addition or a deletion of a nucleotide is stuck in there, and it changes the triplet code now is going off of a different triplet code, and it may lead to, so now it's transcribing off kilter, and it's going to reach a stop codon somewhere nearby, unlikely, it's going to likely reach, it may go on for a while, and then it's going to stop. And that's called a frame shift. Again, that's another type of loss of function. Splice site, here we have no P dot. We have a C622 plus 1 G to T. So there's not going to be a P dot with this one. We have to look at the C dot to see what's going on. It's probably affecting splicing. And then deletions, also, we have to look at the cDNA level. It's an addition of one or more nucleotides. Here, we deleted one, and here we added one, deletion, insertion, and it's at the cDNA level. And then a duplication is when a whole chunk gets copied and makes a duplication of itself. And all of those have downstream effects that are a little uncertain. But those are the different types of mutations you'll see. And the gain of function ones are always the top one. Some loss of function ones are also the top one. Pedo is also a missense mutation, but he seems to be, well, he's neither gain nor loss. But there are other missense that can cause loss. But then all of these are likely to be loss of function. So the loss of function category has a whole host of things that can cause it. The gain of function, it's really restricted to just these single amino acid changes. And here, the 1768, because the channel is opening and closing in a very specific way, this particular mutation changes the way inactivation occurs. It prevents inactivation from occurring completely. So that leads to excess sodium flow and hyperexcitability. So that's a missense, and it causes a gain of function. So we know of a lot of gain of function variants, and we know of a lot of loss of function variants. So I've got them shown in different places. These are all gain of function, and these are all loss of function. So now we're in the process. You can see some of these are found in multiple individuals. These are likely missense variants. But you can see, yeah, the ones that are in blue are missense variants. The ones that are in open or white are either frameshift, nonsense, splice, or indel. So the missense loss of function are in blue, and all the other ones, those complicated ones, are in open white color. Whereas for gain of function, they're all missense. The open ones represent a single individual, and the colored gold ones represent multiple. So does that make sense? So these are all missense. These are missense plus a combination of frameshift, nonsense, splice, and indel. These guys can occur anywhere because they just stop the production of the protein. These guys occur non-randomly. There are certain parts where if it hits, it's going to hit something important, and it's probably going to be a bad outcome. Here, it can go anywhere because it can, at least the frameshift, the nonsense, and the splice sites, they don't care where they are because they're just going to stop the production of the gene.
Yeah.
Yeah, I wanted to ask Michael about the different types, whether some of those may offer opportunities for future treatments that aren't 8A specific, but are about the type of mutation. Like I know there's an advocacy group that's just nonsense variants, and they just work on nonsense variants. So is that like another path where
Yes.
new treatments might be developed?
Yes. So a nonsense variant is a loss of function. It's only producing one copy of the protein when you need two for normal function. So in that case, you may try and up the production of the protein. So it's like in Dravet syndrome, it's loss of function variants. It's an inhibitory network. So you lose one function, but you get a gain of function like effect. So In the case of Dravet syndrome, the gene therapies are going to try and up the production of SCN1A because it's a loss of function disease. In the case of a gain of function disease, you want to go the other direction. You want to knock down the expressivity of that gene. And so you have a whole different approach gene therapy wise. So if you're focusing on nonsense variants, you're going to try and find methods to increase the production of that protein to a normal level. You don't want to go beyond the normal level. That's the problem. That's why you need biomarkers to say, well, we're producing the right amount of protein at this gene because we have biomarkers that tell us it's in the right range of expression. You don't want to knock it down too far if you're going from a gain of function problem. For the same reason, you can get to a loss of function disease if you knock it down too far. So very tricky to get it expressed just right in these different gene therapy approaches. Those are mostly ASOs that do that. And we could talk about that in a separate network meeting about all those different approaches and how it depends on what type of variant and what type of disease it is. We happen to be a gain of function on an excitatory neuron system or a loss of function on an excitatory neuron system, whereas Dravet syndrome is loss of function on an inhibitory neuron system.
Michael, it also might be interesting for people to understand that our understanding of the makeup and the kinds of mutations that exist and the frequency with which they exist in SCN8A is changing pretty quickly. Like I would say maybe was it 2 years ago, we thought that 10% of our population was loss of function. Now it's closer to 20%, right? So I think, you know, we're constantly learning, we're constantly finding out new things. The more and more that genetic testing, you know, becomes available, especially for different things like autism or, you know, different markers or symptoms, we're going to continue to learn more and more about it. And the more that parents get tested along with their children, and we see maybe more mild versions. So I think it's just, it's really, this is a rapidly changing landscape.
Yeah, that's really true. And I think the reason is sort of in this slide, because in the gain of function, 100% have seizures, and in the loss of function, only half have seizures. So if genetic testing is biased towards people who have seizures, you're going to not test as many loss of function. But now loss of function, we know, has delayed speech and autism and motor coordination issues. Well, doctors may not have been very keen to test individuals with those as their primary symptoms. But now genetic testing is getting more likely to occur, I guess, for whatever reason. That's a whole interesting sociological aspect to this. Like, why do doctors decide to genetically test an individual? You don't have seizures, you've got something else. So that's why we're discovering more and more loss of function. And who knows what percentage of the total population that it will account for. It might be 50%, it might be more, because people with autism haven't been genetically tested, or people with delayed speech or ADHD generally aren't tested. But seizures, you are tested. So anyway, this just shows you in a very simple word cloud sort of way that the features of gain and function and loss of function are quite different, but there is some overlap. There are artistic features on both sides, developmental delays on both sides, hypotonia is on both sides, but it's to different degrees. So some of our families, like Pedo, has a combination of these issues. Chloe has a combination of these issues. So we know that their individuals, for one reason or another, don't fit neatly into gain or loss. And we don't know why yet. For
keep learning from them. So it's so important that you guys, that's why it's so incredibly important that we, you know, learn from you all, right? That's the only way we're going to continue to understand the full spectrum of SCN8A, is continuing to hear from you guys year after year about what's going on. So And I know the next slide coming up, Suzanne, you were asking about this. So about how do we find out if it's gain or loss?
Yeah, I wasn't necessarily going to go into that, but if you want to do it in the laboratory, it's an expensive test that you don't usually order. You have to know somebody who works in a lab that you convince them to take your variant, like that Stephen and Kristen just did for pedo. Some guy at Michigan State took the DNA sequence and made a cell and tested the properties of the cell. And that was done because the researcher was interested in the question, but it wasn't the doctor that ordered the test. It's just not a test you can order. But basically, it's complicated, and you can look at the electrophysiology of the cells to see if it's in the cell that you've made is letting more sodium in or less sodium in, and then they determine whether it's gain or loss of function. But we've come up with a way to do it by just using a model where we take a truth set. A truth set is a bunch of individuals that have had this thing done, and then we look at their symptoms, and then we take those that have loss of function and known symptoms, and those with gain of function, known gain of function and known symptoms. And then we say, can we just look at the symptoms alone to predict whether what the laboratory test would tell us? And we've got a paper coming out in Neurology Genetics that shows how to do this. But taking a whole bunch of symptoms or phenotypes, like a history of seizures, age at onset, whether they were motor seizures, whether they were mild or severe intellectual disability, and then we plot those individuals in 3 dimensions, and it's called the principal components plot. All the loss of function individuals wound up below the plane, and all the gain above the plane, and there's a little tiny bit of overlap. And those overlaps are the people who are not quite sure. But we have 90-some% accuracy in predicting whether they're gain or loss, given our truth set. So that means that a clinician could take a patient who presents with a certain set of symptoms and say, plug it into our model, and then get a probability of it being loss or gain. And in most cases, that probability will be, again, it'd be like uncertain or likely or for sure. And so then you don't have to worry about that test, and the doctor can say, I'm not going to prescribe you a sodium channel blocker or what have you. So our predictive modeling approach is, and we're doing the same thing with gain of function to see if there are different gain of function categories. And I'm going to talk about that in future meetings. One of the students in my lab, Josh Hack, is doing predictive modeling just on gain of function. And it turns out there's 3 categories of gain of function: mild, moderate, severe. And he can predict them in his model with fairly high accuracy, just based on certain phenotypes, certain symptoms. And then we're going to see if those 3 categories of gain of function correlate with particular medications that work differently, or different variants that would predict whether you're going to be mild, moderate, and severe. And the answer to that is yes. We've done some work where we can predict mild, moderate, severe. I don't have it here, but these recurrent variants, 850, 1475, 1617, 1872 W and Q, and 1877, we have enough individuals in our registry to see if they predict or if they're associated with different severity of outcome. And the answer is yes, statistically yes. And that's what we're publishing now. We're writing a paper right now.
Michael, can I ask, you've written it so that it can be used by doctors, but can you go through it sometime, obviously not today, so families themselves could apply the test?
Yes, yeah, we can try and go through it and tell you what all the features are, and we can you can check boxes to see which side of the spectrum you're on, the gain or the loss. Yeah, that would be cool. I'm trying to get somebody to make a web app where you could just go in and click on a website and get a result. We have to get a student or somebody who can make a graphical user interface where you put our model in the background, you click, let's say 5 boxes, and then the model runs in the background and it spits out a probability of gain or loss. That's the goal, to try and get that done.
In the works.
in the works. I don't want to go too much further because we've really covered a lot of ground, but next time we'll talk about, it's too bad this slide kind of messed up here, but if I make it smaller, does that help? Yeah. So we think that there's a way. I'm going to try and make all these things over here not so big. Oh, shucks. Try it again. Anyway, it's hard to see that there's two sides of the coin with loss and gain, and the loss is associated with neurodevelopmental delay without epilepsy or with generalized epilepsy, which is generally absence seizures. And then there's a third missing class that might be associated with myoclonic seizures on the loss of function side. And on the gain of function side, it goes from benign epilepsy to mild, moderate to severe. It's just kind of a spectrum, and certain variants correspond to each of these. So 1877, for example, is going to be on the mild side, 850 is on the severe side. And over here, most missense variants are in this direction, and truncating variants tend to be a little more severe. So we're trying to sort all this out. I won't go into it now, but this is one of the things we're going to is coming out in our next paper. And I just leave it as a teaser so that we can talk about it in another network science series chapter. And also what's coming out in our next gene reviews article, and this will be also part of the clinical guidelines at some point. We have the gain of function here. It's just another way of showing what I showed you before, but then we're going to try and associate the seizure types that go with these different 5 groups. The age at onset, the motor development, speech development, cognition, and other. This is our overall goal, is to nail down what these phenotypes are associated with, what the features are associated with these phenotypes, and then try and associate that with a type of variant. And this will help with the clinical guidelines process. And I don't know if Jayed or Gabi want to talk a little bit about that, where you guys are at, but that's all I had for today. I know that's a lot, so that's a teaser for the future talks.
Thanks, Michael. A lot of good, a lot of love in the chat. I also placed a quick survey that should take you like 1 minute to do in the chat. I'll place it again here in case people are able to do it, just so we can continue to tailor what we're doing to what families most want and need. So if you're able to, fill that in, that would be amazing. I am happy to give a quick update on a few other things. One being the diagnosis and treatment guidelines. And essentially that is, we're about 75% of the way there on that. Our next step is that we are kind of processing the round 2 of data, which is where we kind of sent out questions and statements to about 30 pediatric epileptologists around the world to ask them if they agreed with certain statements that we came up with about SCN8A. It's this scientific process called the modified Delphi, which is really about building consensus. And so the next step is that we are going to have one last survey that we send out to both clinicians and family representatives to help us hone in on what we're going to be able to say we know about SCN8A based on the research. And I think a little spoiler is that doctors don't really understand SCN8A. I think probably all of us know that from a lot of our appointments. Some doctors get it and they read all the research and they understand it. I think for the most part, though, there's still a lot of education that we need to do with clinicians around what we're learning from the registry. And one of the most important things with that is the publications that Michael and his team have been working incredibly hard on really taking all of the data over the last 8 years and processing it and putting it together so that it can be analyzed And we are very confident that the registry, the publications coming from the registry, the education that the Alliance is going to be able to do with clinicians is going to help move us forward. So as long as people are able to continue to contribute to the registry, that's going to be the game changer for our community, really. We know that after the guidelines are complete, the idea is that we're going to publish those in a journal, and they will then be open access. You know, it's not going to be behind a firewall where people are going to have to pay to get them. We're going to make sure it's printed so that anyone who gets diagnosed can find that article, print it out, take it to whoever does the whether it's a pediatrician or a geneticist or a doctor that does that testing, and be able to say, this is what we know about SCN8A. My kid has this kind of an SCN8A presentation, and we know these are the kind of meds that are going to work best. This is the kind of therapy they're going to need, and that will help to advocate. I get emails from people all the time saying, we're having a hard time getting therapy. We're having a hard time getting resources. The idea behind this is that once it's published, it's in the public domain, and it becomes evidence that we can use to advocate for better care and treatments for our kids. It's not going to have all of the answers, we know. And so the International SCN8A Alliance is working with our global partners. I'm not sure if people know, but we partner with SCN8A Italy, SCN8A Netherlands, SCN8A China, SCN8A, you know, groups around the world. And as leaders of these groups, we're going to come together and work to develop a global research priority kind of list of things that we think are the biggest gaps in SCN8A that we want to prioritize researching. And that's going to build our agenda for the coming years, right? And so it's going to be important that we can rally the entire community around these kinds of things. And we're going to need to do fundraising to fund research, because this is going to be very targeted. We're going to say things like, we know that SCN8A, there are serious GI issues across the board, right? That's not something that clinicians are identifying. That's something that our families are identifying. But we need to be able to document that, do the research, document that in articles, and then it becomes, again, public knowledge that is out there, and we can take it and say, prove it, right? We can prove to our doctors, this is an issue. We need you to take it seriously. We need you to help us get better treatment and get references and resources. Okay, I see, Mariana, I'm going to slow down. So that's really, really an exciting advancement for our group. We know that by the end of this year, we will have these guidelines published. They will be a resource so that people who get diagnosed can get the best possible care and treatment as soon as possible, no matter where they live. And then it's going to lead us to this really exciting next step, which is that as a global community, we're going to come together to say, here's what we need to prioritize learning more about for SCN8A, both in the labs and within clinical care. Did I forget anything, Jayetta or Michael, that you want to highlight there?
No, you covered it well.
I'm just responding to Joanna, who just wrote that she'd love to help, and we need help. So Joanna will be in touch, but anyone else, this is really a complex effort. We work on so many fronts. So if there's any way you want to get involved, give us a call, jot us a note, and we'll find a way that you can work, because there's so many activities, and it's only going to happen the more we have families pitching in and making a difference. So anyone who has ideas, just a little bit or a lot, everything's going to help.
Yeah, and there'll be lots of opportunities. I think one of the biggest things is going to be that we're going to have a very ambitious research agenda that we want to get funded. So it'll take us all working together to try to rally our communities around it. Unfortunately, with so many rare diseases, you know, we are pursuing grants from NIH, different research bodies, to try to help us move these things forward. But the more that we have a community standing behind us and helping us to advocate and to raise funds to do an ambitious research protocol, the more successful we'll be, the faster we'll find answers. And I know not everybody can give. We don't want anybody, our lives are complicated. We're all stretched. We're all trying to pay the bills. So we're not asking families to give beyond what they want to, but it's also just about helping us tell the story. It's really important that we can share the experiences of those living with SCN8A. Some of you have shared your stories, your experiences. We'll be reaching out to ask for those again to help us really rally a community around us. And frankly, we're looking to start doing some major gift, looking to get some bigger donors in the door. And the only way that happens is if they see what our lives are and how we can move that forward. Zipporah, we definitely need you. We can learn together. So thank you for stepping up. And I'm touched because honestly, I know several other people on this call who have already stepped up and helped in other ways. And that's how we'll get there together, right? Tammy, everybody knows Tammy. She's out there working the lines and finding people and connecting people and answering questions. Carolina just hooked us up with somebody who's going to try to help us with some of our website issues. So it's like, it's really just about us rallying together to find ways of building something stronger, faster together. So we're just really grateful for this community. You guys are amazing. I see we have some newer families who are joining us today. I'm so glad you can make it. I won't call out your names, but thank you for joining us. Reach out if you have questions. I'm sure you do. Let us know how we can support you. I know Sharon, you had, oh yeah.
So I want to know if people want specific questions answered in the next talk. Send me your ideas, your questions. How should we be doing that? Should we use scn8a.info for questions about that? Do you want to have it go through the Alliance?
Yeah, I think we'll keep serving people through our newsletter and through our Facebook group.
I apologize for we have a complicated landscape with, multiple websites and social media groups. And it's kind of there's a lot of history to that, but we're trying to pull it together and make it simpler. But if you do have questions for me specifically about your genetic report or any genetics or anything like that, you can always find me at scn8a.info@gmail.com. And Elise and I go through the emails every week. We try and stay up to answering your questions on a weekly basis. So anyway, again, if it's more general, Gabi, you suggest ways people go through a more general sort of route.
Mm-hmm. Yeah, people can always reach us. I'll drop my email in the chat. People can find me on Facebook. I'm not as good at Facebook, but always reach out because we want to hear from you. And Zipporah wants your jacket. We actually do have SCN8A. Michael, you want to model it up close? You want to show people? So we just made these fleeces. And I'll put a link to the order form in the chat. We are happy to, that's kind of one of our fundraising elements that we're putting out there. So Stephen's got one. Yeah. And they're really, really warm. JS got them here. They are really warm. So we're happy to, I'll put that in the, I'll drop it in the chat in a minute, and then I'll also drop it in our Facebook group.
I'd also like to ask Chris, I don't know if Madeline's with you, but I covered some areas that you guys are going to be more expert than I, and I would just love for you guys to contribute to one of these science network meetings, ask your permission for that. And if there's anything you wanted to add or to clarify at this point.
Thanks, Michael. I really enjoyed your seminar. Great information. Nothing to add, but we would be happy to participate in the future if you'd like.
Absolutely, especially on the gene therapy side.
Great.
On the what side?
Gene therapy and the splicing and all that stuff that Chris and Madeline are expert in.
Yeah, and there's also been some interest in splice, which I know that, Chris, you are knee deep in your lab and an expert in. So that might be something that we can dig into for families a little bit more as well. Is splice site variants.
Yeah, I'll be happy to talk about that. Just sort of general background on splicing and
yeah, I think that would be great. Sharon, you've had your hand up for a while. What's up?
Hi guys. I'm just always like curious. As you know, Zach, dad has the SCNA mutation, and I know he was affected in other ways. So my question is like in the future room for you guys to do research on people like Jose who has the SCNA mutation, but to kind of get him as a guinea pig to kind of do research as to what protected him. You know what I mean?
Mhm.
That kind of stuff. It's my question that I think, like, to me in my mind is like that's kind of like the key, kind of finding out these people that weren't affected, like what's their defense mechanism, you know what I mean, that in their DNA, that, you know what I mean, that's where I'm coming from, because I think that will be key in the future, you know.
Yeah, we do have some projects that we're working on where we're looking at combination of variants to see. We have one family that has both gain and loss of function, and it actually segregated as one generation got the loss and the next generation got the gain and the loss. So we're making cell lines from those individuals. It's tricky to think of an experimental design, like in your case, Sharon, where we could get Jose's DNA and do something to understand. Sometimes it takes making a mouse model where you put each variant separately in different mice and cross them, but that takes time and energy and effort, and money. So we have to be very careful about what kinds of approaches we take, but those might be the kinds of research priorities we come up with as we go forward as a community, and then try and raise the funds to try and support scientists to do that kind of work. I think that's going to be our best bet. Modifiers, I love the whole thing of modifiers. Like I said, I've been working on it for years, and I would love to understand more about SCN8A and how it can be, that gene itself can be modified in various ways through, you have to find a pedigree. We are actually working on some inherited families right now. We're drawing up the pedigrees where several generations, just like the one I showed you, are passing on the same SCN8A variant. It's a very mild one, but there is some variability in how family members are affected. And in one case, there was a more severely affected, just like I showed you on that family pedigree, and we might be able to figure it out as to what's causing that. Is it environmental? Is it medication-wise? Some of the family members were on one medication, some were on another. What associates with the severity of that particular family? It's you know, story after story like that will eventually fill in these gaps in our research.
Thank you guys.
Sure
Amazing
thing.
work.
And Julia and Cam are deep in the research game too. They, if people don't know, they raised a lot of money to have a study going on sick kids in Canada, looking at potential biomarkers. So just calling you all out on that. But what was your question?
Oh, it was actually just in terms of, and sorry, we missed a lot of the first part. So I have two questions. One, was it is it the recording something that'll be shared out? Because we were away. Okay,
Yep.
cool. That was my
Yeah, I'll
first
share it
question.
later today, probably.
Nice. And then my second one was just, you know, in the variant calls that we used to have it, like for 1617, you'd see kind of like their growth trajectory of like from a development standpoint. And now that we have, hopefully people updated the registry, like, is that something maybe Dr. Hammer we could see in some future meetings, is based on the updated registry?
Yes.
How are like those specific variants doing?
Yeah, so again, the topics we were going to cover, this was my initial jotting down this morning.
Yeah.
This was one of the topics that I thought was really important, is to look at the skills that kids develop, and depending on which variant they have or what have you, and why that is, why earlier seizure onset can be more disruptive to acquisition of developmental skills in terms of the critical periods that children go through when they develop between the age of birth and 2 years old. So there's a lot of information on that for other epilepsies, and we've got some information on that for our epilepsy. And we also see differences depending on variant. So that was one of the topics, and we will definitely cover it in one of these meetings. Some coming up.
And then just last question too, as we were talking, and I know the sick kid study is going on. Like as we think about like the potential biomarkers, are there biomarkers that exist right now for SCN8A, or like that you're aware of too, or like how does that work?
Jayetta just is coming back fresh from a meeting, right?
You're muted. You're muted, Jayetta.
All the epilepsies are way behind other disorders in the development of biomarkers. And biomarkers are kind of physical changes in the body that can help diagnose, identify better treatment, identify outcomes. I mean, it's really like the what's it called? The golden ring or whatever. The answer to almost all our questions is the development of more reliable biomarkers, but it's a nascent field. It's not going to happen quickly. There, aside from your effort, you know, most of the work is being done broadly in the across all different types of epilepsies. But again, in our research priority conclave that we'll be sponsoring, I mean, that's our whole point, is to together as a community globally with all the partners around the world and families really target what those priorities areas are and then fundraise around it, grant writing or you know, finding researchers to work with to promote application for existing grants. I mean, there's a huge biomarker grant program at NIH, and we would just have to put the package together. So I think we're really excited about this priority setting of research because, you know, there are 1,000 directions to go, and all the amazing work that Michael does, that's mostly not funded. Michael does this on his own, and rigorous, comprehensive research requires funding, and we need to get more funding for Michael because he's the one really leading the field in getting answers about 8A.
Yeah, that makes a ton of sense. And hopefully when the results come back for those who have participated so far, because I think there are definitely some people here, then, you know, hopefully that'll help progress. But also this was awesome and very exciting, all the work that the Alliance is doing. That was it. Thanks.
We're also, you could say that we have an effort to make a 16,
Yes.
17 mouse.
Yes, there's an effort to make a mouse. So thank you to Dr. Hammer. So also looking to see.
Yeah,
if we
we're
can
going to
make
be
another
doing it
mouse.
here at the U of A. We got a really good quote for making a 16, 17 mouse. It'll take us 6 to 9 months, but it seems like we've got Manoj Patel is going to be able to fund it. So we'll have another mouse model with another variant. So we'll have the 850, we'll have the 1617, the 1872, the 1768. There's some loss of function mice. So yeah, all good stuff.
Perfect. And then someone just asked, just while I was here, who said it's like to for the oh, never mind. Sorry.
20 grand.
Oh, good.
20 grand to
Mhm.
make the mouse and to dispense it to the laboratories that'll study it.
Yeah, it's it's costly, but you know, this will be hi Madeline, hi Margot, hi sweet girl.
Michael, you should say though, it's more if it's a severe strain. The 851 took like 5 years to get.
Well, that was tough. The
That was-
1617 one should be better, easier.
Right.
But there's a conditional mouse and then there's a straight knock-in mouse. And we're going to make a conditional mouse anyway, even if 16, we don't know if 1617 in the mouse will be tough. I'm thinking it's not going to be, but we're going to make the conditional mouse anyway.
Well, I'm saying that Tammy asked you how much, and I think you gave the answer for the run of the mill, not
No,
one
no,
of
that's,
the.
well, we got a good quote. So I don't know
Right.
how much it would cost to make a conditional mouse. Other places it might be more. Maybe Julia and Cam remember what some of the quotes were, but
Yeah, they were significantly.
a
more
company
than
was
that.
like 50 grand at a company, but the U of A GEMCorps, I mean, it's a nonprofit institution. So I think that helps.
Yeah, we're also working on a project, hopefully, to better map where all of these different mice are, where all these different models are, right? So there's a lot of different ways that you can do research in a genetic disorder. So there's mice, that's one way of developing a way to study, modeling what it would look like in mice to try to understand what it looks like in humans, which of course is not perfect. There's development of pluripotent cells, which are, I shouldn't be the one describing this, but basically they're taken from a human, so it's a sample from one of our kids, which I think probably a number of us have done this. And then they grow these cells in plates to run testing on and model things. Somebody else can take over talking about the different models, but there is an 1872 mouse, right? We have
Yes.
1872.
Miriam made a conditional 1872 tryptophan W. There's not a Q.
Yeah.
And we showed with our work that Q and W are different. Q is milder than W. And there's zebrafish. There's what else, guys? There's and iPSCs, mice, zebrafish.
And then you go to bigger animals once you have, and those are mostly for drug testing often, right? But
then there's another whole really painful area to talk about, but that's very fruitful, and it's brain tissue after children who've passed. And we've actually been working with the University of Maryland and have a brain tissue bank dedicated to 8A. And we have a webinar that we did on that. And that is one of the most powerful ways to actually start learning more about premature death. So it's a tough topic. I know Gabi and John struggled with it for years. She was committed 100% to do it, and it still took a long time to complete that form and make that commitment. But it never ties you down. You do the advanced education, and then at the time, you still have 100% freedom to make a choice about it. But that's a really powerful area of research. I know we all worry about premature death, and the only way we're really going to learn about it, or the most promising way, is from brain tissue from those who passed.
Yeah, and just to clarify a little bit, so the idea here is that if you think that you might be interested in having your child's brain donated when they pass, if they pass,
Just a tissue.
No, it's the brain. They take the brain. And what they do is, so the idea is that there's a lot that you can learn from actual human tissue brains where this disease is occurring, right? And I put a link in the chat that we had one of the leading scientists who does, development of studies from tissue that is collected. And so, we have some FAQs on there. Our partners at Hope for Harper do a lot of work around brain donation. And so we can answer a lot of those questions, but the idea really is that this is an incredible way to, have a legacy for our kids to continue to help us learn. And I'm happy to talk with anybody who might be interested. You can sign up now and do paperwork. And then if you don't call, if something happens, then they don't ever know and they don't bother you. So it's just doing the paperwork in advance in case you might be interested. So we have families who have been through brain donation that we can also probably have you connect with if you're interested in it. There's some webinars, or reach out to us. And I just also want to note that Cam put an update in the about how you can get engaged in this biomarker research if you're interested. So go ahead and grab that email there. We can post it, you guys can post it again to the page if they're still recruiting. Feel free to drop that in the on the caregiver page again too. So
yeah, for sure. We were going to do that. We were supposed to have a meeting with them, but the doctor has been sick for the last couple of weeks. So we're waiting for that, find out an update, and then post it. But I think we should.
Wonderful.
I should mention that we've done some studies of brain tissue that have been resected during surgery from temporal lobe epilepsy patients, humans. And I can talk about that in another science series talk. We do the transcriptome, so we sequence all the RNA from the resected tissue of these patients with temporal lobe epilepsy. And that may sound like a different kind of epilepsy, but we find amazing similarities between mice that have SCN8A and humans that have temporal lobe epilepsy. So I will talk about that in a future meeting.
Dr. Hammer, I'm not sure if you saw that article, someone posted it a couple times in Australia, like that there was some university that was studying drug refractory epilepsy in Australia, and that they were studying and they think that they might have a cure or something like that. I've seen it not posted on a couple groups. Are you aware of that?
No, I'm not.
Okay, I'll post it in the group.
Okay.
Yeah, anyway, it seems like it's far away, but anyway, just wondering if you knew.
Yeah.
Yeah, and Megan asked in the chat about connecting to people who studying different variants. I mean, that's part of the reason we want to start this kind of not a bank, but like a warehouse or like a log of who's doing what research and has what models. So, you know, I mean, if you wanted to find something, you look it up and you can see what public papers have been published, who's got what. You can reach out to us, but frankly, there's really only like 5 or 6 variants that are really mostly being studied because that's what models are available, you know, so it's 1872, 1768, 850. What's the one that just got made by the woman from Miriam's lab? I'm just blanking on her name.
760.
What is it? 716?
767.
767, right. So that's where most of the research is concentrated. And that's why, you know, it's important to have additional models being made to really represent. We all know that SCN8A is a lot of different disorders, right? It's like most of our kids don't look alike. And so like, it's almost, you know, like there's really a very, very wide range of SCN8A presentations and phenotypes. Some of the work that we're doing with the diagnosis and treatment guidelines is identifying and getting consensus that there really are 5 distinct types of SCN8A. And Michael is verifying this also through the registry, that there really are these distinct types of SCN8A. And the more that, and this will be a part of our research agenda planning as well, is like, where are we missing the mark, right? What are we not able to study? And that's why we were really, really big advocates for the development of this 850 mouse that was made in a lab down at Emory by a guy named Andrew Eskig and his team, because if you can't study what's going on in more severe kids, you can't learn what drugs are working, you can't learn what kind of impacts there are from seizures, anything. So it's really important to have these different models. You know, Chris and Madeline are working on models in their labs to help us better understand the spectrum. So I think, and this is something that Madeline shared after she went to this big genetics meeting in Europe, is that SCN8A is just a really amazing model for understanding epilepsy. So I think the work that we've done over the past 10 years as a community to get out there information about SCN8A, build models, is paying off, because people are starting to see SCN8A as this strong model for epilepsy. And that means more people are going to be studying it, and we'll learn more and more. So I don't know if Madeline, if you want to say anything about that, or Michael.
Yeah, I mean, I'm trying to convince the post-traumatic epilepsy, the TBI community, get interested in SCN8A, because it is, if there's any kind of brain injury, because of the place where SCN8A is expressed on the neuron, the nodes of around VA, those regions of the neuron get disrupted easily in accidents, concussions, military guys in the field, football players, all kinds of people who get brain injuries through that kind of physical activity likely have SCN8A involved, and nobody knows this. So it's something we're trying to promote. I'm writing some grants. It's very tough to get people to think outside of the box, But if in fact SCN8A is a key player in these other forms of epilepsy, it will become a big focus of research. So we're trying to get that accomplished.
Any other questions or thoughts? Yeah, Tammy.
I'm not sure if it's showing my hand up because I don't know how to do that, but
Well, the real hand works too.
do I got to press on it or
No,
something?
no, you're fine. Go.
Okay. My first question is, are on the 850 mice, are they actually displaying the same symptoms that our kids are having, or is it different? Does anybody know?
I think it's too soon to tell. They made the mice, but now they have to breed them, and because it's such a strong mutation, you can't just have the F850 expressed. You have to turn it off and then turn it on at certain stages. And I think they're still in the process of figuring that out.
They did share. I mean, the reason it took 5 years to make is they couldn't get these mice to stay alive. So they had to use this very specialized process and modeling to make it work, which is this turn on, turn off thing. But they did, I mean, they came and talked to an 850 meeting at one point, and they talked about a lot of serious issues that they saw in the mice, like not being able to move very well, like having feeding issues, right? They said a lot of the mice died because they couldn't get them to eat. Well, we know that probably is just like in our kids. They have severe, motor issues and probably aren't able to suckle well enough to get enough sustenance to survive. So I think they are probably going to be able to do that, but we're not sure yet. Yeah.
And then my second question is, the researcher here in Michigan, has anybody gotten a hold of them or?
Not yet.
Okay, because I can load Liam up and go down there. It's like a half hour from me.
What about Dr. Hernandez? Shouldn't Dr. Hernandez be able to link us up?
I don't know.
I
was talking to Kristen about that. I
know, Kristen. Well, Kristen, you talk with them, right? You're part of their study. Can you write to them and say, can you please contact Michael Hammer to?
So I'm not in contact with them.
Oh, you're not? Okay. They just took a sample?
Yeah, everything is through Hernandez. Sorry, the boys are playing, so it's loud at my house.
That's okay. It is Saturday after all.
It is. But Hernandez said that he's not at MSU anymore. He's at the hospital that he works at, which is now named Corwell, because they just combined. So I took the screenshots and sent it to Michael and was like, this is everything I found. And then it was like, wait a minute, he's not there. So I don't know if that's it.
Okay. Well, we can keep digging. Michael, I can help try to follow up on some of the information that was sent. We'll see if we can track them down. I mean, I think it makes sense for us to be collaborating and sharing information. I'm guessing they could learn a lot from the registry and comparing some notes. So thanks for trying to make those links, Kristen.
Anytime.
All right. So any other questions? I know we're over. People are being generous with their weekends and their time. I will post a recording of this if you want to send it to other people or watch again because you missed part or for any other reason. It will be a general resource for the community. So share it with anybody, doctors. I know, this information probably is less relevant for doctors, but yeah, and what we'll do is for our next meeting is we can put out some surveys to try to see where people are most interested in going next. And we're grateful for your time. Thank you, so many of you, for raising your hands and saying you want to help. We will follow up with you because we are eager to work as a full community to finding answers together. So appreciate that. And if any of you have, lingering questions that come up, feel free to reach out to Michael or I, and we'll try to get you some answers. And we'll see you on these next meetings. Again, if you don't have it, you can always go to our website at SCN8A and click on the upcoming events, and you can find we have meetings just about every week. And frankly, you are welcome to join any meeting. Even if it doesn't really seem like it's probably, relevant to your kid. Tammy comes to just about every single meeting and learns and then uses that knowledge to be, in the community and helping families connect and make linkages. And you might come up with some other ideas about you know, what might work for your kids. So you're always welcome. Oops, that was the wrong link. I didn't mean to put the brain bank link there. What I was going to do is I was going to put in the link for the survey. If people want to share any, you know, any feedback about the meeting. Oh, it doesn't want to let you do that. Then we're, you know, that is also what we will use to try to start, you know, thinking about how we continue to do these meetings. If you liked it, you know, if you have other topics that you want to hear about, and if you have feedback, you know, we're always looking for testimonials so that people can see the work we do at the Alliance. I think there's still a lot of people in the SCN8A community that don't really know about these meetings or what we're doing, or so please try to help spread the word. And if you're willing to share a testimonial, that would be amazing. And I'm going to stop there and let everybody go. Thank you all so much. We'll talk to you soon. Be in touch anytime. Enjoy the rest of your weekend. Bye, everyone.
Bye.